Hello, I'm John Moult, a computational biologist at the University of Maryland in the US. In this segment of the course, I will discuss some aspects of variation in the human genome, setting the stage for subsequent segments on how variation contributes to different types of disease. The genome of each human contains approximately 30 to 100 variations from the reference genome, not found in either parent. These result from errors in DNA copying and DNA repair. This error level is such that in the US, with a population of about 300 million, the implication is that each mutation is introduced de novo several times per generation. The longer-term fate of these variants is a complex combination of purifying selection, random drift, the exponential growth of the human population. Some variants will be sufficiently deleterious that they will rapidly disappear. Others, even though deleterious, may persist for many generations, particularly if the disease impact is recessive, requiring both copies of a gene to be affected for a disease phenotype. Some may drift to a high frequency in the population, especially but not only those that are relatively benign. As we will learn in a subsequent segment, some of these contribute to complex trait disease. The 1000 Genome Project has provided a comprehensive view of the amount of variation in typical human genomes. The largest number are single nucleotide variants, called SNVs, over 3 million in a typical European genome. Although the number of insertions, deletions, and copy number variations, CNVs, is smaller, these actually cover a larger number of bases. Variant data extracted from sequence information is usually compressed into a so-called VCF file. These files contain a list of the variants detected, together with critical information on the reliability of each. As you can see from the slide, there is a lot of information. It's important to realize that just because a variant appears in a VCF file does not make it real. All current sequencing technologies have random and systematic errors of different sorts and frequency of occurrence. Although the sequence information, such as read depth and strand bias, can be used to estimate reliability, in practice, an alternative sequencing technology is often used to validate variants of clinical interest. A graphical view of the data using a genome browser helps a user to quickly estimate reliability. The figure shows one example, a single base change in the MECP2 gene that introduces a stop codon, so causing a truncation of the corresponding protein and producing an intellectual disability phenotype. The pop-up box provides access to genotype quality information. In this instance, the variant is already known to be pathogenic. The type of serious disruption of protein function seen in the MECP2 gene is called loss of function, LOF. Besides the introduction of stop codons, as in this case, other types of LOFs are frame shifts, loss of stop codons, disruption of splice sites, and larger insertions and deletions. We will see in later segments that particularly for monogenic disease, the most frequently observed potential pathogenic mutations are single nucleotide variants that change an amino acid in a protein. These missense mutations are harder to interpret. Many computational methods have been developed to address the problem. Most are based on analysis of a multiple sequence alignment from different species. We learned about alignments in an earlier segment of the course. The basic idea here is that if a mutation introduces an amino acid inconsistent with those seen in the alignment, it's likely to be deleterious. In the example shown, the reference sequence and all the other species included in the alignment have a charged arginine residue at the mutation site, while the mutation is to a bulky tryptophan residue. From a physical chemistry perspective, this is likely damaging to protein function. A quantitative estimate of how likely the mutation is to be deleterious is achieved with relatively straightforward machine learning methods that take a set of known disease-causing mutations and known non-disease-causing mutations and learn what features of an alignment are consistent with each. If this seems a crude approach, it is. The reason these methods achieve some degree of usefulness is that most pathogenic missense mutations have a large impact on protein function. As we will see, for complex trait disease, this is often not the case, and their variants that affect gene expression, as well as subtler missense variants, play a major role. Some analysis pipelines may supplement the sequence method results with inspection of analysis of the three-dimensional structure of the protein, if it's known. As we will see later, that also has the potential bonus of yielding insight into the underlying molecular mechanism. In later segments of the course, we will discuss the role these germline variants play in monogenic, cancer, and complex trait disease.